Rios. I'm research fellow for the University of Glasgow, and we just finished to moderate to start the session of the trainee advocacy in inflammation and immunity. So we just start this open discussion about what you found what you just found interesting in the presentations, and also what's the take-home message about this. I would say for Dr. Pitzer.
Yeah, so I'm a postdoc at Vanderbilt University Medical Center, and our lab really focuses on salt sensitivity of blood pressure and how the immune system plays a role. And right now we have a human study where we are looking at these isolevoglandins in antigen-presenting cells. And the reason that we're doing that is because there's not necessarily a specific marker for a diagnostic marker for those who are salt sensitive. And it's very difficult to have patients come in, give them a high salt diet, and then salt deplete them the next day. So if there's a way to do a blood marker test of who is salt sensitive and not, that is the purpose of what we're doing, is to see perhaps if the inflammasome within these cells or isolevoglandins within these cells can correlate with the level of salt sensitivity of blood pressure, and use that as a diagnostic marker for patients who are coming in with hypertension.
That's very interesting. I was reading about salt-sensitive hypertension kind of just last week, and I actually got very interested because salt also can accumulate in the skin.
Yes.
So do you think that these dendritic cells in the skin, Langerhans cells, would play a different role? Or would play also some inflammatory conditions as well in terms of salt sensitivity hypertension?
That question is, and it's a huge question that is not answered, is where are these innate immune cells encountering these high salt environments? And so the question is it in the skin? So when there is a high sodium concentration within the skin. So if you have these antigen presenting cells, T cells that are, going into the skin, experiencing this high salt environment, that could be a possibility. So part of what we're actually doing with these patients is we are doing skin MRIs. We're doing MRIs to see where is the sodium in these patients.
So I'll go now for Dr. Shokoples. What do you find more interesting in the data and the presentations?
So I'm actually not a doctor yet. I'm still a doctoral candidate, but okay. Thank you. So yeah, with my project, we were interested in, so we know the immune system plays a role in the development of hypertension. Everyone here is familiar with that, but we wanted to look for an early trigger. And so clinical data has shown us that there is elevated ATP in hypertensive human patients, and that patients that have loss of function mutations in P2X7 are at decreased risk for the development of essential hypertension. Mm-hmm. So for our data, looking at the P2X7 knockout mouse model of hypertension, we're able to show that there was a decrease in only systolic blood pressure, which was particularly interesting. So this could provide a treatment for isolated systolic hypertension if this holds true with a P2X7 antagonist. And we're also able to protect against endothelial dysfunction and, to some extent, vascular remodeling.
Yeah, that would be my question. Where do you think that those ATP receptors would be more important, in the endothelial cells or in the immune cells?
It's a very tricky question. And one way that we've went about to start answering, which is still early in the process, is doing an adoptive transfer. So we irradiate the mice and then we adoptively transfer them with either wild type or P2X7 knockout immune cells. And hopefully that should answer whether it is, immune mediated or endothelial or something else.
Mm-hmm. Okay, so they believe that ATP works as a dump, danger-associated molecular pattern, because it's quite clear there in the data. And then this dump, they go. Where would they be more important? Would it be the blood circulation, or is it actually the tissue that increase the inflammation in the vessel, or in the blood circulation that we interact with endothelial cells and start the whole inflammatory response in there?
Yeah, that's a very good question and not easy to answer. I think the answer is both. So I think in circulation, it could activate endothelial cells, which then can recruit immune cells, further amplifying inflammation. And then same within the tissue, it could activate immune cells causing local tissue inflammation, which self-amplifies the cycle.
Mm-hmm. And what do you know about a condition that we can find increased ATP in the kind of extracellular ATP? Because ATP is mostly intracellular. So what's a condition that you find the most extracellular ATP that this ATP can actually bind to the receptors extracellular in the membrane?
If we're talking about acute instances, tissue injury, so if any cellular damage, and more chronic, there's been some evidence that blood vessel stretching, like endothelial cell stretching, can cause ATP release that could trigger activation of immune cells through P2X7.
Okay, that's interesting. It's so many things about ATP that we do not know yet.
Yeah.
About those receptors that people actually are working on it all the time. And I'll go now to Dr. C. So please, can you tell us a little bit more about your work and what you found interesting?
Yeah, thank you for having me today. And also, I'm still a PhD candidate, so I'm not a doctor yet, but thank you. So my work focuses on understanding how the gut microbiota regulates blood pressure, and that's actually the focus of our whole group. We have been digging into a novel perspective, pH sensing, which is important because many cardioprotective metabolites produced by the gut microbiota, like short-chain fatty acids, are acidic. And I think the two major take-home messages from my talk today are, one, the cardiovascular protection by acidic metabolites derived from dietary fiber is partially through their acidic nature. And two, an anti-inflammatory proton sensor, GPR65, contributes to this protection, likely through inhibiting certain pathogenic inflammatory responses in hypertension, such as type 1 cytokine productions. So I think my work highlights a protective immune suppressive role of acidity and may apply to broader application in the control of cardiovascular diseases.
It's very interesting. So do you believe that different diets actually reduce or increase, will have the different influence on the pH?
Yes, absolutely. For example, for dietary salt, we believe it will change the pH in the gut as well. But we haven't got the data yet, but we have already applied for an ethics to perform experiments to try that. But we assume it might cause metabolic acidosis in the gut, and that's reducing the pH. But we think this mechanism might also be a checkpoint to control the inflammation.
I see. So if you talk about, because you're very, very focused on cardiovascular disease, and it's kind of understandable. However, you know that because the diet and the fibers and those pH changes, the pH are also very much associated with some of this colon disease, inflammation, colon inflammation. Do you know anything about this, about the change in the immune regulation, the immune system in terms of Th17 or CD8 T cells, or all those, or the immune cells that stays there in the intestines?
That's a very good question. So we did have the flow cytometry data from the gut, but we haven't analyzed all the data yet. We have the data from the control mice, which are not angiotensin 2 infused, but we did not see any significant difference between knockout and wild-type mice. But from the literature, we do know that GPR65 is a determining factor for inflammatory bowel disease, and the knockout mice indeed developed exacerbated colitis in several models. So we think there should be some physiological change and inflammation in the gut.
Okay, and where is mostly expressed this GPR65? Do you know?
Yeah, so that's also a good question. So GPR65 is widely expressed in almost all the immune cells, but it's more highly expressed in the T cells, and it's also have another name, TDAG8, because it was first found influencing T cell death.
And sorry, just very curious. It's just about what would be the main ligand for GPR65?
That's a very complicated question now. So there was reports saying some lipid might be the actual ligand for it, but it is quite controversial and even caused some retraction. So the actual ligand for GPR65 is unknown, but it's quite clear that pH can change the activity of this receptor profoundly. Because Proton is so small and it's not likely to be a conventional ligand for a receptor. So the investigation on the actual ligand for this receptor is still needed in the future.
So let's discuss a little bit, Shobana, please tell me a little bit more about your work and the main data, and how do you actually see your data?
Yeah, hi all. So I'm from Texas A&M University, and my lab focuses mainly on the role of renal lymphatics in the regulation of blood pressure. So I did my PhD in reproductive endocrinology. So somehow I was interested, and I browsed through the literature available, and I found that there were only limited papers which actually dealt with the effect of hypertension on reproductive function. So there were few papers which stated that there is a reduction in the hormone levels and alterations in the testicular microvasculature. But I couldn't find any paper which actually describes the molecular mechanism behind the reproductive dysfunction which are observed in the hypertensive patients. So I somehow came up with a hypothesis that macrophages play a role in it. Since macrophages, especially the pro-inflammatory macrophages, are increased in the several end organs like kidneys and brain and all. So I was interested to see if hypertension has any effect on testicular macrophage polarization. So we just started with the flow cytometry, which was supportive, and we saw an increase in the M1 macrophages in the testis of hypertensive mice. And followed to that, we saw inflammatory markers and a reproductive dysfunction in the testis of hypertensive mice. So this is all we got as of now. So we'll be planning more experiments to elucidate the actual molecular mechanism.
Actually, yeah, but you found beside macrophage thing, the macrophage function, you actually showed to us several very interesting data about lymphangiogenisis.
Generally, our lab has few papers which shows that hypertension induces inflammation-associated lymphangiogenesis in kidneys. So even I followed the same, and I found there was a significant increase in the lymphatic density in the testis of hypertensive mice. So we found that there was an increase in lymphangiogenesis in the testis too.
Okay, what would be mostly autoimmune disorders? Do you know which kind of, such as one example of autoimmune disorder that could be most related to angiogenesis in the testicular?
It's generally experimental orchitis, which is the inflammation of a testis. So that's the most that's a widely used experimental model for autoimmune disease.
So that's very good, guys. So thank you very much. Thank you really much for today, because I then soon have to start another round of oral presentations to see